In today's class, we shall discuss about the pigment chlorophyll. From our last class, we came to know that the chlorophyll pigments are present inside the chloroplast. Now, specifically where the chlorophyll pigments remains present. If you have followed our previous class on the structure of chloroplast, you will came to know that inside the chloroplast there lies the thylakoids. Inside the chloroplast there lies the thylakoids, and the thylakoids piled upon one another to form the grana. If we observe the ultrastructure of a single thylakoid, then we will be observing the thylakoid is also double membrane as that of the chloroplast, and inside the inner membrane there are small dot-like structures are present. These dot-like structures are called quantosome. And if we observe the ultrastructure of quantosome, it consists of three layers. The outer layer contains carotenoids, the middle layer contains chlorophyll B, and the inner layer contains chlorophyll A. So, the pigment chlorophyll remains present within the thylakoids, more specifically within the inner membrane of the thylakoid, or more specifically within the quantosome. So the pigment chlorophyll remain present within the quantosome in its middle and the innermost layer. Although in nature different types of chlorophyll molecules remains present, but generally when we consider about chlorophyll, we generally talk about two pigments, that is chlorophyll A and chlorophyll B. Beside chlorophyll A and chlorophyll B, other chlorophyll pigments like chlorophyll C, chlorophyll D, bacteriochlorophyll are also present. But generally we consider the two most abundant form of chlorophyll, that is chlorophyll A and chlorophyll B. Now if we look into the basic chemical formula of chlorophyll A is C55 H72 O5 N4 Mg, and that for chlorophyll B is C55 H70 O6 N4 Mg. That is, if we compare chlorophyll A with respect to chlorophyll B, the differences are in the count of hydrogen atom and in the count of oxygen atom. Remaining things like carbon, nitrogen and magnesium are constant in chlorophyll A and chlorophyll B. In chlorophyll A, there are present two extra hydrogen atom, whereas in comparison to that, chlorophyll B bears one extra oxygen atom. Now, if we look into the ultrastructure of chlorophyll molecule. So, this is the basic structure of a chlorophyll molecule. The chlorophyll molecule consists of four pyrrole rings. This is ring 1. This is a pyrrole ring. This is ring 2. This is ring 3. And this is ring 4. So, chlorophyll molecule consists of four pyrrole rings. And why these rings are called pyrrole rings? Because these pentose ring have nitrogen as one of its constituent, which is why it is called pyrrole ring. Now, beside these four chiral rings, it also contains an magnesium atom at the center. This magnesium atom holds the entire structure together because it is linked with all the nitrogen atoms of the chiral ring. Due to the presence of four chiral rings, the structure of chlorophyll is called tetrachiral structure. The structure of chlorophyll is called tetrachiral structure. And due to the presence of magnesium atom at the center, the greenish coloration of chlorophyll pigment is exerted. If questions come, why chlorophyll is green in color, the answer will be due to the presence of magnesium ion at the center of the molecule, chlorophyll is greenish in color. This type of porphyrin structure is also present in the hemoglobin which is present in our blood. The only difference is that instead of magnesium ion, in case of hemoglobin, there remains presence of iron, which is why it is red in color. So in general chemistry, magnesium is greenish in texture, which is why the chlorophyll pigment is greenish in color. Similarly, the iron is reddish in texture, which is why the hemoglobin is red in color, as within the structure of hemoglobin there lies the iron atom. Beside this four chiral ring, a fifth ring, ring 5, also remains present, but it is not a chiral ring. Instead, it is considered as pseudo ring. Why this fifth ring-like structure is not a chiral ring? Because it lacks the nitrogen atom. Beside this, it has been formed by association with ring 3, which is why it is considered as pseudo ring. Now, if we again recall the structure of chlorophyll A, which is C55 H72 O5 N4 Mg. So we can see here there are 4 nitrogens and 1 magnesium ion. The 4 nitrogens are the part of 4 chiral ring, and the single magnesium ion are present at the center. Now, if we compare this molecular formula with the molecular formula of chlorophyll B, what we will observe? In case of chlorophyll B, lacks 2 hydrogen atom. Means in comparison to chlorophyll A, there are 2 hydrogen atom less in case of chlorophyll B, and 1 oxygen atom more than that of the chlorophyll A. Remaining formula is same. Now, where comes this difference in the number of hydrogen and the number of oxygen, where the difference exists in ring 2, in ring position 2. In case of chlorophyll A molecule, in this position there will be methyl group. That is, if in the structure of chlorophyll at ring 2, if methyl group is present, then it is chlorophyll A. That is, methyl group signifies chlorophyll A. But if in the same position aldehyde group is present, if aldehyde group is present, then it signifies chlorophyll B. So the basic structural difference between chlorophyll A and chlorophyll B lies in ring 2. If it is methyl group present at the ring 2, it will signify chlorophyll A molecule, and if it is aldehyde group, then it will signify chlorophyll B molecule. Now just compare the functional groups. In case of chlorophyll A, there are 3 hydrogen atoms, while in case of chlorophyll B, there are single hydrogen atom. So if we subtract that, so there are difference of 2 hydrogen atom. This 2 hydrogen atom is lacking in chlorophyll B, which is why in comparison to chlorophyll A, chlorophyll B has 2 hydrogen atom less. Similarly, in case of chlorophyll B, we are observing a surplus oxygen atom, which is absent in case of chlorophyll A, which is why in the molecular formula there is one additional oxygen atom present in case of chlorophyll B in comparison to chlorophyll A molecule. Now, beside these phytol rings, the structure of chlorophyll molecule also contains a lipophilic tail portion. A lipophilic tail portion. This is the tail portion, which is lipophilic in nature. That is, it is hydrophobic in nature. Hydrophobic in nature. While the above head portion, which constitute the 4 phytol rings, is hydrophilic in nature. So the structure of chlorophyll contains both hydrophilic portion as well as hydrophobic portion. So it is preferred to extract chlorophyll using 80% acetone, because 80% acetone will have maximum extraction power to extract the chlorophyll pigments from the biological samples. Now coming to the dimension of chlorophyll molecule, the porphyrin ring, the dimension of the porphyrin ring means this upper head portion is considered as the porphyrin ring. The dimension is 15 into 15 Armstrong or 1.5 into 1.5 nanometer, while the dimension of the phytol tail, that is the tail portion, the molecular formula of which is C20H39, that is the phytol tail consists of 20 carbon atoms, is 20 Armstrong or 2 nanometer. meter. If in exam questions come, which chlorophyll molecule lacks the phytol tail, means this phytol tail portion, this hydrophobic portion, so the answer will be chlorophyll C. Chlorophyll C is the molecule that lacks the tail portion. Besides this, also remember the color of the pigments. The color of chlorophyll A is bluish green, while that for chlorophyll B is yellowish green. We know that beside chlorophyll molecule, some accessory pigments are also present within the structure of quantazome that act as light harvesting pigments. Among them, the two most important are carotene and another is xanthophyll. Now, what is the structure of carotene and xanthophyll? Carotene are nothing but are typically 40 carbon containing compound that is composed of 8 isoprene molecules and are probably terminated in a ring. That is, carotenes are nothing but have a molecular formula of C40H56. So, carotenes are terpenes that consist of 8 isoprene molecules and are terminated with a ring. While in comparison to carotene, xanthophyll are also a type of carotene that also contains 40 carbons along with the oxygen molecule and are considered as oxygenated carotene. Now, if we study the pigmentation color of carotene, it will be orange, while that for xanthophyll it will be either yellowish or yellowish green. So chlorophyll molecule is a cyclic tetrapyral structure. Now, if in exam it was asked that name a molecule which is linear tetrapyral, that is chlorophyll is a tetrapyral structure that we all know. But the tetrapyral structure, that is the 4 pyral rings are present in a ring-like structure, so it is a cyclic tetrapyral. Now, if in exam question comes, then name a linear tetrapyral. Linear tetrapyral pigment. What will be the name of a linear tetrapyral pigment? It will be phycobilin. Phycobilins. So in our next class, we shall study about effect of light on photosynthesis.